And we come to the next presentation by Noam Harei from the University of Minnesota, Minneapolis, High-Resolution Imaging and Anatomy.
Thank you, Carl. So my role is actually talking about some high-resolution imaging and how we can actually get these images. So I slightly changed the title, and I'm actually talking about exploring the brain at high field. And when I'm talking about high field, I'm talking about 7 Tesla and above. It should be a Mac. Oh, yeah, here we go. Here we go. Okay, so let's have a quick introduction of what resolution actually buys us. So here we have the same data set in which we actually was downsampled to 8 by 8 by 8 volume, each of the voxels, 4 by 4, 2 by 2, and 1 by 1. Initially, of course, the 8 by 8, you know that you can see maybe it's a brain, but as you go to smaller and smaller voxels, you can see more and more details. So initially, it's not that striking, but when you're trying to compare between the 2 by 2 to 1 by 1, that's an 8 times more difference in SNR when we compare between the two. So you need 8 times more average to get from the 2 by 2 voxel to a 1 by 1 in order to get the high resolution. So everything boils down to SNR because the imaging, the signal is proportional to the volume that you're actually imaging. So the whole idea is going to high field, you actually get higher SNR. That allows you to actually push the resolution to smaller and smaller voxels. So when you go to high field, you have 2 main benefits. Of course, one is the SNR, the signal-to-noise ratio, that I just explained. But then you have also increase in the contrast. And here the contrast, I break it down to 2 parts. One is the anatomical contrast that allows you to distinguish between 2 different types of tissue, and functional contrast, and that is more relevant when we're talking about fMRI. And throughout the talk, I'll actually try to explain that. So I'll break my talk to 4 different parts. The first part will be talking about high-resolution vascular imaging, how we can actually image the vessels, because if we're trying to actually push the resolution for fMRI, we have to understand what's the underlying vascular system, because everything is the plumbing underneath. And the second, how we can actually use these information to try to push the fMRI to try to get some high resolution. The second part of the talk is more focused on anatomical aspect using imaging with 7 Tesla. So we're talking about how we can actually distinguish between different parts of tissues, and of course, how we can actually use that to push the clinical aspect for fMRI, for anatomical. So let's start with the vascular part. So what we have here, we have most of the studies that we did, the development was actually done with anesthetized cats, and this was done on a 9.4. And what we have here is a high-resolution T1 weighted. So it's an axial view looking on top of the surface down to the brain, and we have coronal view running back and forth across the images. And what we see, we can actually see these dark lines coming in and out as we scroll through the plane. Now these actually are vessels, and they are veins. And how do we know that they are veins? Because they're darker than the tissue. And that's because of the high concentration of deoxyhemoglobin. Just think about it as a static bold signal from the tissue. So Pat Boland, who was working with us at the time, said, you know, this is only half of the story. You can actually get high-resolution venogram, but we're missing half of the probably more interesting, which is the arterial side. So to image the arterial side as well, Pat actually combined the time of flight or the inflow effect. And how do you do that? You actually position a slab just below the surface vessel, which is running on the surface, and you saturate all the spins within that slabs. And what does it do? When you have new blood, which is fully relaxed and is rushing from the surface, diving deep into the tissue, it will actually show up as bright. So in the same slab, we have vessels which are bright, and you can see it in three slices here. Or you have vessels which are dark, which these are the veins. So we can actually go in and tag each one of the vessels that we identify, both as arteries and veins. And then if you're trying to get the bigger picture of how the vessels are, you can actually do some post-processing with some post-processing and with subtractions, and you can actually remove the surface vessels and look what's happening to the vessels below. And now you can actually track each one of these vessels, and you can combine that with information of what type of vessel is it, and then you can actually generate a three-dimensional vascular model. And what does it buy us? This is done in vivo. So now we can actually combine that with fMRI and try to figure out what's the spatial relation between the vessels and the arteries, or the veins, basically. Very similar finding was actually done with ex vivo from the monkey model with Bruno Weber in the 2008 work, and which with a much higher resolution, but you get very similar spatial organization of the vascular tree. So as I said, now the whole idea is to try to combine the two together. So we have the vascular system in these cats, and we have, in this case it was visual simulation, and you can actually see the ball signal is a function of how it's related to the vascular system. And all of these are intracortical vessels. We're not talking about large surface vessels, but we're actually talking about vessels which are diving into the tissue. Okay, so if we switch on to fMRI, so the whole idea, of course, is how we can actually map the brain when it's active. So this is images from the first classical Ogawa paper in '92 with the first fMRI. So you can see the quality of the anatomical imaging and the blob of activation that they basically identify as functional signal, the ball signal. So as we know, as we try to progress and try to develop more and more or better tools to actually use the fMRI, of course, one of the reasons is actually going to high field. This is a nice study by Issa Yacoub in the early days of the 7 Tesla, in which he actually scanned the same subject on a 4 Tesla and on a 7 Tesla. And you can see how significant the activation maps are actually when you go into a 7 Tesla. But when you look into the signal change, you see a significant changes for the 7 Tesla. And that's the CNR, the contrast that I was talking about from a functional perspective. Because now you can actually be much more sensitive to smaller changes for the ball signal. This is when we're still talking about gradient echo. But when you go in and you start using spin echo, people started to notice that the signal is not actually coming from the surface, but it seems that it's actually following the gray matter. And you see somewhere in the center of the gray matter, you have a hot spot or the larger activation for the ball signal. And of course, the question is, where is that coming from in the tissue? Because if we're trying to understand where what the fMRI signal is representing, we have to understand what's the underlying anatomical corresponding for that. So this was actually displayed already during the talk. So the whole idea is actually, we know that the cortex is organized in 6 layers, which defined by different the cell size, type, or density. The same idea as Brodmann used to actually define these areas. So the question, can we actually extract this information and try to relay that to the anatomical information that we actually get from the fMRI? So once again, we go back to the cat when we did the relatively high resolution fMRI studies, 150 micron in-plane resolution. We see the anatomical image on top, and we have both gradient echo ball, that's the standard ball signal that everybody's using today for fMRI, and the spin echo, which is typically more useful when you go into high resolution or in high field. And what we see that in both cases we have, if we just look at the tissue, so if you can just follow the gray matter and the white matter here, you see that in both cases you have some kind of line of an area with high activation in the tissue. And the question is, where is that signal is coming from the histology part of the visual cortex? So what we did in these, here we go. So in several of the cats, after the imaging session was done, we actually sacrificed the animal, and we had to find the corresponding anatomical area that correspond to the image, the part that we actually imaged for the fMRI, and we actually did the histology, and what we did, we actually used the exact same criteria, and now we actually identify each one of these layers based on the same histological criteria. And then we can actually go back and reconstruct these areas, but now we can actually know exactly where the borders of the layers based on the anatomical definition for each one of these slices. So once again, we have the fMRI map, we have the anatomical stuff, and then we actually plot the ball signal change as a function of distance. So we have from the surface down to the white matter. So from the surface to the white matter, from the surface to the white matter. And what we see, even in 9.4, that the strongest signal is actually coming from the surface area. But that's not surprising. If you just look closely on the map, you can actually see how the strongest Bolt signal is actually following the surface. Well, that's not surprising, as I said, because if you go back to the vascular studies that we did, and we try to correlate the two, and we can actually see, we'll take a slab, a MIP across the area that is roughly corresponding to the area that we did the map. You can see that the majority of the power in the image is actually coming from the surface. And you can see very nicely, actually, these intracortical vessels running into the deep of the tissue. So going back, we can actually see that indeed, even in gradient echo Bolt, 9.4 Tesla, the strongest signal is still coming from the surface. But if we eliminate the surface area, we see that somewhere in the tissue, we got this second bump. These are 3 different cats. And that bump is always around layer 4. So when we do the same thing with spin echo, 2 interesting observations. One is that you actually eliminate or reduce the surface non-specific activation, and you can see also in the map. But more interesting is that when you're looking at the tissue, the peak is actually occurring once again over layer 4. So as we know, the layer 4 is the main input area for those regions. So how we can actually combine all of this know-how that we developed from the animal studies to try to push the human fMRI stuff. So from the early days, we get these blob of activation, which didn't tell us too much more than what we knew in the past about this correlation between brain function and localization. But what we really want to know is we have to develop a method in which we can actually push the resolution and try to map not these millions of neurons that are actually active, but actually the thousands of functional units, these cortical columns, that we know that are probably part of the basic computational units of the brain. So if we're trying to understand how the brain is working, we have to at least map to the level of the columns. A quick introduction of what cortical columns are. So probably the most studies are the visual system, the ocular dominant columns, in which if you record from cell, so this is a chunk of cortex from the visual cortex, we have the surface white matter and we have the 6 layers. So if we record from starting from the surface perpendicular to the white matter, you'll find cells with similar response properties to one of the eyes, whether it's right or left eye. And the whole idea is that these are actually organized in a column-like fashion. This is why they are called columnar organization. So we can actually have columns of cells that have similar response properties. But if you look on the surface, it shows as if it's a zebra pattern of alternating between left and right. So the first hemodynamic-based mapping of ODC was actually done during the 80s with optical imaging, in which you actually do a craniotomy, you shine a monochromatic light, and you stimulate the monkeys, and all of this is being done in vivo, and you stimulate the monkey, one eye at a time, and then you can actually do a subtraction between the 2 images. So this is the picture or the image that you get from the surface of actually before the stimulation. And during the stimulation, you can actually do a subtraction of eye that images were acquired while one eye was presented or stimulated at a time. And you can see these bands of activation of left, right of activation. And this is once again based on hemodynamics. So that's ODC with optical imaging. And that was, of course, very nice, represented by the 2DG, which was that at the time was the gold standard of actually mapping functional activity. But of course, this is a one-shot because you have to sacrifice the animal and do histological afterwards. Hemodynamic based, you can actually repeat these experiments again and again because you're not changing the brain. Another nice example here is that you can see very nicely the border between V1 and V2 because V2 doesn't have the ODC. So that's another nice explanation or example of how we can actually get anatomical information from the functional properties that we're actually imaging. So of course the goal was to try to push that to the human studies, and over the years people use the ODC as the benchmark for actually how we can actually push and test the fMRI in humans. So initially it was done with ex vivo, of course, and then starting in the late 90s, and you can see the progression of the functional maps of ODC that people did over the years. So we did the same thing in Minnesota using the 7 Tesla, and this is work by Issa Yacoub. And the whole idea was actually following Kang Cheng's approach, in which you, by choosing a very specific slab slice just perpendicular, actually parallel to the calculable feature, you can actually optimize your chance of actually seeing this alternating pattern. That's exactly what Isa actually showed. You can actually see left, right, the preferences. So this is nice, but this was shown already, and you have, for ODC, you have an anatomical basis for this separation between the two. Another set of cortical columns are orientation columns. So if you record from cells in visual cortex, and you present stimulation with different type of orientations, you can actually find neurons with preferred orientation of the stimulation. So initially, when these type of cells were actually identified, Hubel and Wiesel actually got the Nobel Prize for identifying all of this stuff. But in the initial days, the concept was believed to be that this was actually organized as ODC and orientation, actually perpendicular to each other. So they came up with this ice cube model at the time. So once again, so optical imaging came to the rescue, and this is data from cats, anesthetized cats. The green bar represent the orientation of the stimulation that was presenting to the animal. Dark patches represent the areas that respond stronger, so that's specific orientation. Now, initially, it looks like as if it's just random scattering across the brain. But once you combine the frames together, you can actually see the pattern that is emerging here. You can see that you have a representation of 180 degrees surrounding a specific point. So it has some kind of radial arrangement for the presentation. So Greenwald and colleagues actually assigned different color codes for each one of these orientations, and they came up with the concept of pinwheels. So you have a singularity point in which you have a representation of 180 degrees surrounding that specific region. And that was a very nice example how imaging can provide new information that wasn't available at the time, which was contradict the electrophysiology. At least when you get the bigger picture, you can actually resolve things much better. So we set to actually do the same thing in humans. So we had the same subject line in a 7 Tesla, and we presented a rotating bar, and we binned together all those voxels with similar phase information, and basically generated a map that looks very similar to the optical imaging. But the question is, how can you actually validate that? Here you can just poke an electrode and record the electrical activity. In this case, we had to actually build the case based on what was known from the animal studies. But the most obvious feature, of course, is the pinwheels. So if you go back to the data and you plot the data as a function of time, so it's actually following the phase information, and you see that it's not just randomly scattered across the brain, but it's actually following some kind of a pattern. So if we can actually use a monochromatic and speed it up, and you can see the pattern that actually is emerging here, which is very similar, of course, to the pinwheels that was shown with optical imaging in cats and monkeys. So another nice feature is that for the same subject, we have the ODC. So now we can actually superimpose the ODC on the phase information and look at the spatial relationship, and there's a whole literature between the two, between the ODC and the orientation columns, and it's actually following very nicely all of what's known from the monkey organization of orientation and ocular dominance in monkeys and in humans. So overall, fMRI today, if you use the right tools, you can actually reach and map to the level of cortical columns, and I think Rainer will actually talk about that later on today, which they actually took it to the next level, and it's actually in area MT, and showed columnar organization in areas that wasn't known that that's existing. So Rainer will probably talk about it later on. So the last part of the talk is actually more focusing on the anatomical features and how we can actually get more information when we actually go into high field. So this is the exact same patient that was scanned on a 1.5 and on a 7 Tesla. And you can clearly see the differences between the two images. So it's not only that you can actually see better features or it's cleaner image, but you can actually extract more information. So the beauty is not actually matching the 1.5, but actually pushing the resolution, because you have much more SNR to burn, as we call it, to actually push the resolution and get smaller and smaller voxel or get more and more information. So you can get very nicely information between, for example, in the white gray matter, of course, but you can actually get nice information within the gray matter, and you can actually follow very nicely vessels running in the soul site. Now, when you go into high field, you can actually start using or taking advantage of sequences that are more advantageous when you go into high field. For example, SWI, stability weighted images, which take into account the phase information. And Jeff Dunn from NIH actually did some very cool work with all of this stuff. And he showed that you can actually get and extract much more information to the level of layers based on the phase information that you can actually extract from these images. So clearly, by taking advantage of the phase information, you can actually enhance the contrast or the ability to distinguish between different features in your image. So these are the same subject line, 7 Tesla magnet, classical T1, T2, and SWI. And what you can see that if you focus in the brain, in the central of the brainstem region, you can see that T1, even on 7 Tesla, is relatively flat. You don't have too much contrast. On the T2, you can see very nicely the STN and SN, the nigra and subthalamic nuclei, and you can see the red nuclei very nicely. But look how much more information you get when you're actually doing the SWI. So you can see the head of the STN, you can see the tail of the nigra, and you can even see features inside the red nuclei. So once again, you can actually get more information just by changing the contrast of the acquisition. Now, each one of these acquisitions, we're talking about a 5-minute acquisition. It's not, 30 minutes acquisition with a grad student lying in the magnet and not moving. These are actually patients that you can actually get this type of information in a 5 or 6 minutes acquisition. This is a coronal view of SWI, and a nice example of the contrast increase is you can actually see the separation between the STN and the nigra. If we'll just zoom. So you see it's not only the resolution that actually buys you, but it's also the difference in the contrast between the two that actually buys you the information that you can actually extract. You can see nicely the borders between the GPI and GPE, the globus pallidus, internal, external. So when you have so much information from these type of images, you can actually go back to the Bible of neurosurgery, the atlas, the ex vivo atlas, and try to figure out how well you can actually do in vivo today. So on the left, we can see that it's a slice through the globus pallidus. On the right, you can see the GPI and the GPE. And if you can actually superimpose the atlas, you can see not only that the borders between them, but you can actually see the lamina between the two. So it, once again, buys you a lot of information where you can actually push the resolution. So by taking all of these different type of images, the T1, the T2, the SWI, we can actually today reconstruct a truly patient-specific anatomical model for the patient that we're actually imaging. And why do we need that? Because the whole idea is actually you want to move away from atlas-based and you want to actually move into patient-specific anatomical targeting. Because when we're doing surgeries for neurosurgery, you want to actually image or you want to go to that specific target in that specific patient as opposed to going to that target based on some other people's map. So the whole idea is actually doing this patient-specific images. So this takes you to the last part of my talk, is how we can actually use all of this information from high field to actually do some clinical work. So the project that we actually chose is actually deep brain stimulation, or DBS, and the whole idea is that mainly for Parkinson patient of movement disorder, that medication basically doesn't help anymore. The whole idea is today is to stimulate electrically the brain by actually inserting an electrode into a very specific target in the brain. The electrode is hooked underneath the skin with a wire, and you have a pulse generated, looks like a pacemaker. And the whole idea is that you need to get into a specific target, at least for the movement disorders, the majority of targets is the STN, the subthalamic nuclei, and you have to get with an electrode and stimulate that specific region. Now, you have to get the electrode into the right location because two things. One, if you won't stimulate the right place, the patient won't get the benefits, or worse, the patient will actually get side effects because this is prime real estate location. We have multiple targets all around it. So how DBS is being done today in the clinic, since you need to go deep into the brain, you have to use images. So these are the clinical images, a 1.5 or 3 Tesla. in which you basically don't see the target area. So what we do today, we actually superimpose an atlas. This atlas was derived from, I think, one or two old French ladies that donated the brain for science. The brain was sliced, and based on that, today we're still trying to figure out the location of the target based on those two French ladies. Now, there's, of course, as expected, there's a variability between the location of my STN and Carl's STN and so on. And the whole idea is that how do we find the STN today? So the procedure today is actually, in most cases, going with microelectrode recording, MER, and the whole idea is you actually map as you advance the electrode. So each different nuclei will actually have a different firing pattern. Once you hit the STN, hopefully you will recognize that and you know that that's in the target. So this is a long process. Typically it's between 5 to 6 hours, multiple penetration, and the patient has to be awake doing this procedure. Why? Because if you anesthetize the patient, all of these lines will basically be flat. So you won't have any MER information. So you cannot validate where you are from a targeting perspective. And once again, the success of DBS is crucially dependent on how well you can actually stimulate or position the electrode. So today we scan patient in Minnesota on a 7 Tesla, and for each one of these patients, we actually generate this atlas. So the yellow represents the STN, the target location, the blue is the Niagara below it, and just as reference is the red nuclei. And what we can see, we have 6 different patients, and we see that it's not exactly the same shape, size, or orientation. And the whole idea is that we need to find these locations during the surgery. And don't forget, today what we're doing is actually using one coordinate system to try to match for these different patients. And the whole idea is that one size fits all doesn't apply anymore. We reached to level today that we, on a per patients, can actually figure out exactly where we need to go. So during the surgery, we actually generate these type of models. We go to the OR, we show the neurosurgeon or the neurologist, the guy who's actually doing the mapping. This is the target location. This is where you need to go. This is the orientation. These are the borders. So during surgery, that's very useful. After surgery, we'll take a post-op CT, and then we can actually merge the two together. And what does that does for us? It's actually give us indication exactly where the electrode yeah, here we go. That gives us indication of exactly where the electrode ended up in the target. Because you have all of these patients, you have patients that respond beautifully when you turn on and off, and you have patients that don't respond. And the whole idea is why, what's the problem? And today we have this information because we can know exactly where the electrode is and what contact of the 4 contacts that today are relevant for stimulation are in the right location. So all of this is, once again, bringing the entire story into a whole, in which by doing that you can actually take the images, use it both for fMRI and both for the clinical side, and actually help progress. I just want to acknowledge people who work on multiple projects here, and if you have any questions, I'll be happy to answer. Thanks.
Any questions? Yeah.
Sorry, so just a basic question. You showed both spin echo, fMRI, and gradient echo. I thought in the fMRI you want to catch the mesoscopic part of, R2 star, so R2 prime, so to speak, right? So why do you want to do spin echo then?
The whole idea with spin echo is that it's less sensitive to the large draining veins and more sensitive to the small capillaries.
Right. So it's really it has some admixture from R2 prime as well, right? Otherwise you wouldn't see anything. So then it depends on the echo time, right?
That's correct.
So does it depend strongly? Does it varies from site to site? Is everybody using spin echo at say 15 milliseconds? And
so we use the same echo time across the brain,
right.
But I know that people are using different echo time for different institutions. That's correct.
And do you plot the intensity, or you determine the rates? these maps are signal intensities with Spinnaker, or are they the actual rates? You take a log divided by t, etc.
No, those are intensities.
So you've
Correct.
okay.
Correct.
Because
That's raw signal change.
I
That's
see.
the whole idea. No manipulation. That's to show you how much the signal is changing as a function
I
of
was just
depth.
wondering whether it's better to do this or to normalize to.
We kept it as simple as possible because once you're actually doing normalization, then you introduce in errors, and then you introduce in bias.
Okay.
We just try to keep it as simple as possible.
Choi from Seoul National University. I have two questions. So for the resolution for the functional MRI, so for the board, can we actually like target a specific nucleus of like hypothalamus or something like this, so resolution and other limits? And the second, is there any computation limits that we require or certain high performance computers to do this kind of bold analysis using 7 Tesla functional MRI?
So regarding the second question about computers, it's exactly the same thing, whether it's a 1.5, whether it's a 7 Tesla, it's the same analysis from that perspective. Regarding getting fMRI from deeper structures, it's harder because it's deeper, so it's actually further out from the, further away from the core, so the sensitivity is decreasing. But once again, it's, you can still get very nice activation. So we have nice activation, for example, of thalamus and so on.
Like sub-millimeter resolution functional MRI board?
In deep structures, you mean? That's pushing it. That's pushing it, yeah.
Hi, Noam.
Hi, how are you?
This is Edson from Brazil. Just a quick one about the distortion when you plan for surgery. Of course, you're not using any registration to any normative that's set, but still you have the distortions that.
So that's a
have to
great
be
question.
corrected.
So that was the question number one that we had to address before we actually can take that to the clinic. So we had a study in which we actually took 12 patients that were scanned on a 1.5 that went through the entire process of the DBS as we do in it today. And these patients were actually scanned on a 7 Tesla as well. And then we looked at the registration between the 1.5 and the 7 Tesla. And the whole idea, we're trying to figure out if we can use the 1.5 as the gold standard because it's being used today, we have to see how much distortion we get between the 1.5 and 7. And if we are okay with that, we can actually, we are safe. That's exactly what happened. So we have distortion from the 7, and that's not uniform across the brain. So when you go to areas which are close to air cavity and so on, yes, you get higher distortions. But if you're looking at the areas that you're interested in for DBS, for example, that's the center of the brain, you don't have almost any distortion over there. So the distortion is acceptable to the 1.5 distortion that you compared, at least for the CT, which is still considered the gold standard. Exactly.
Thank you.
That's a great question.
Yeah.
Thanks.
So, thank you very much. We have a break, and we will continue exactly at 10:30 here. Thank you.